Can anybody see my screen? Can reply with a yes at the chat room, okay? Hello. Can you see my screen?
Yes, I can.
Okay, all right. Thank you so much. So once again, you're welcome to the webinar on microbial genomics. So where we'll be looking at the research and applications of microbial genomics to health and agriculture. And along the line, we are going to focus basically on an aspect of microbial genomics, and also I'm going to take you through the practical or the workflow, right? The workflow of microbial genomics in terms of metagenomics. All right, this I'm experiencing comment. Okay, is my screen still audible? Right?
Yes, it's still on.
Like, I mean, is my screen, can you still see my screen? Hello, can you still see my screen?
Yeah, we can see your screen.
Okay, amazing. So, I'm Oluwaseyi Olawale. I'm the founder of Genomark Hub, where we integrate creative thinking, innovation about technology to prefer sustainable solutions to contemporary health challenges using bioinformatics, artificial intelligence, and medicinal chemistry. So basically, we explore African medicinal plants, and also we explore microorganisms, where we use this as a powerful tool, right, to prefer sustainable solutions to health challenges, mostly human health. Okay. So I'm going to be taking you through this webinar session. Now, like I said, we'll be talking about research and applications of microbial genomics to health and agriculture. And I believe that this is not your first time of hearing about, microbial genomics, right? Or hearing about genomics. Probably even if it's the first time, I believe that is why this webinar is available, right? That is why this webinar is here, such that you'll be able to have a full detailed understanding on what genomics is, what microbial genomics is, and also we'll be able to look into the research and explore some certain, practical applications, because it is one thing to understand what genomics is, or microbial genomics is another thing to know what area of research are there, right? And what are the applications of microbial genomics to health and agriculture. In fact, when you hear about microbial genomics in agriculture, you'll be wondering, wow, how can you relate agriculture with microbial genomics? Is it really possible? Because if we're talking about microbial genomics to you, I believe you'll be thinking about microorganisms. Yeah, it's about microorganisms. But then, how do you think is relative to agriculture? How can you relate microorganisms to agriculture? You may be thinking about pathogenomics is part of it, but there are some other areas of research, especially when you're exploring the genomics of those microorganisms, in order to understand the relative, the relationship between microorganisms and agriculture. And also, like I said, we'll be exploring health as well. So I promise that this webinar is going to be very, very interactive. Okay. It's going to be very, very interactive. So please, I would like to ask, can you drop at the chat room what you understand by microbial genomics very briefly, so that we can take it from there. Okay, like I said, it's going to be very interactive. I always love my class to be, kind of interactive. So I will be expecting at the chat room, what do you think or what do you understand by microbial, genomics? I'm waiting.
Can I answer, sir?
Okay, go on. Yeah, you can answer.
Okay, I am Dr. Rasha Mustafa from GRC, Desert Research Center, Associated Professor in Genetics.
Okay,
Okay,
go.
I can believe that we are talking about the total genes of each certain microbes or certain species of microbes, and the gene expression maybe of these genes in its pathology for plants or humans. I think that.
Yeah,
Okay.
amazing. Yeah, definitely.
Okay, sir. I
So
thank
thank
you
you.
for this opportunity.
You're welcome. You're welcome. Thank you so much.
Okay.
All right. So yeah. So just like she said, right? So microbial genomics is basically dealing with the entire genome structure of a microorganism, right? So because that is micro, so such that, please can you mute? Okay. We actually wanted to mute everybody, but uh, we have having a technical issue here, so that will be another time. So please try to mute so that we'll be able to follow through. Thank you so much. So microbial genomics, like I said, we're going to be exploring, or it deals with exploring the entire genome of microorganisms. And you understand that microorganisms cut across, bacteria, virus, right? Even fungi, right? And then we have virus, right? Like I said, so how is this relative, right? To, please, um, whose screen are we seeing? Isa Shago. Mutari Sashago, please can you off the camera? Sorry. Thank you so much. Thank you so much. Okay. Sorry. Please can you mute? Oh my God. Okay. So now when we talk about microbial genomics, what do you imagine? Okay. Or in your career, or maybe as a microbiologist, or as any like as a life scientist, but you're interested in, or you have an interest in microorganisms, right? So what is the first thing that comes to your mind? What do you imagine about microbial genomics? What do you understand, or what do you have in mind? What is the research interest you have in mind about microbial genomics, right? So there is a specific research question you desire to answer by exploring microbial genomics as a life scientist, right? As a life scientist, there is a specific research question that you must have in mind, that you must have desire, okay, to answer by exploring microbial genomics as a what? As a life scientist. So, and these specific research questions could either relate to health or relate to agriculture. Because I believe that every research question is a problem, right? The fact that you want to explore microbial genomics could either be you want to explore in order to acquire some information, or you want to explore in order to prefer, in order to, prefer a solutions. Though you can get these information, and these information cannot be used to solve that particular research question. All right, to actually solve a problem. Okay, so, and that is the idea about microbial genomics, and that is the kind of idea that you could conceive, which could be innovative, right, as a scientist or as a researcher when it comes to exploring the microbial genomics. Okay, So it means that as a scientist, you can embark on this particular field of study in genomics in order to solve a wide range of problems that is either related to health or agriculture. Now, understanding microbial genomics is very, very crucial. All right. So if as a scientist you want to embark on this research, or probably you are a PhD holder, a master's student, or even undergraduate student, and you have an idea of, or you have an interest to pursue a research question, right, in terms of microbial genomics, either in health, in agriculture, or even talking about environmental, right? It is very, very understand. It's very, very important, right? You explore or you understand microbial genomics. So now the identification and characterization of genetic material belonging to microorganisms is what we term as microbial genomics. So, and if you're dealing with microbial genomics, there are two things in particular. It can either be pathogenic or non-pathogenic. So it means that when you see a microorganism or when you hear about microorganisms, there are two things are relating to microbes generally. So the first is pathogenomics. The second is non-pathogenic, right? Sorry, the first is pathogenic. So, and if it is pathogenic, all right, if it is pathogenic, it means it is harmful, right? So there are bacteria or there are microorganisms that are pathogenic. For instance, viruses, they are really pathogenic. There are some bacteria that are pathogenic, such that they cause harm, right, to the host. And the host could either be man, like human, right? The host could either be plants. The host could either be, animals, right? In fact, there are some, microorganisms that affect other microorganisms. Okay, so it is very, very crucial you understand what kind of microorganisms do you want to embark on in the basis of your research. Is it a pathogenic microorganism or is it a non-pathogenic microorganism? So non-pathogenic means they are not harmful. And as we proceed, you get to understand examples or areas of pathogenic research and areas of non-pathogenic microbial genomics research. And as you proceed, you now begin to understand, you have a clear picture, okay, as a scientist, or if you desire a research paper, okay, I want to explore pathogenic or I want to explore non-pathogenic. Any one of these, you'll be able to have a clear picture of the workflow that you'll be able to embark on in order to be able to have a very successful research in your career on this topic, actually. Excuse me. Please, can you still see my screen? Just stop. Can you reply the chat room? It's still be. Is it? Can you see it now? Yeah, I believe you should. All right. Okay. So now we have research areas. So let's start from the research areas. Okay. Now, the research areas in microbial genomics, the first is pathogenomics. Now we've understood what pathogenic is and non-pathogenic is, right? So if you talk about research areas, the first is pathogenomics here. And what is pathogenomics? This is the study that encode microbial resistance and virulence factor, which enable a microorganism to infect a host and cause disease or cause infection, right? So the first thing that should come to your mind is this area of microbial genomics research is based on what? Please, can you give me an answer at the chat room? Can you give me an answer at the chat? This is an area of pathogenic, of microbial genomics research, right? This is an area of microbial genomics research, but based on the two categories of microbes, right? Pathogenic and non-pathogenic. So what area of research do you think this particular area, this particular research area is focused on? Is it focused on pathogenic or non-pathogenic? I need an answer quickly at the chat room. That's amazing, amazing. I can see pathogenic, good, pathogenic microbes. Exactly, 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 exactly. Thank you so much. All right. So this is an area of what? Pathogenic. Is this an area of microbial genomics, okay, that deals on pathogenic organism. So that is why it is called pathogenomics. So it is coined from pathogenic and then genomics, okay. So and it deals with this, studying the microbial, genomic structure of pathogenic, microbes, right? And it deals with focusing on the resistive nature of the pathogenic, microbes, the virulence factors involved, and then all these things contribute to enabling the microorganism to infect the host and cause disease. So definitely, if you embark on this research, it is that you are narrowing down or streamlining your research, or focusing on the entire genomic structure of the pathogenic organism to a specific area that is associated to a particular disease, right, or a particular pathogenic infections, right, caused by this pathogen, but by that particular pathogenic organism you're exploring. Now, research has been able to show that certain pathogenic bacteria were able to cause cancer, or are able to actually cause cancer in human, right? So, and then to be able to explore this accurately, it is very, very important that as a researcher, you be able to, focus on the genomic structure through microbial genomics research, right? Focus on the genomic structure of this pathogenic organism, whatever that you have known to cause cancer, right? And then you will streamline your, research to specific genes of interest or specific proteins that are acting or that are expressing the virulence nature of the pathogenic bacteria to cause, the cancer. So if it is infections, for instance, please can somebody give me an example of pathogenic infections at the chat room? Very quickly. Very quickly. An example of pathogenic infection. I said I love my class to be very interactive, right? Okay, somebody said syndrome, hemolytic uremic. Somebody said COVID. Yeah, good one. Good one. Skin infection. And skin infection is caused by what? Somebody can drop it at the chat room. Skin infection, right, is caused by what pathogenic organism? What pathogenic organism is caused by skin infection? Yeah, we have E. coli. That's the pathogenic organism. Yeah, Staphylococcus aureus. Staphylococcus aureus. So Staphylococcus aureus cause, a specific skin infection. Staphylococcus aureus, right? Cause, it also causes skin infection. Now, that shows that Staphylococcus aureus is a pathogenic bacteria, and for for Staphylococcus aureus to cause this skin infection, right, or cause the skin infection, it shows that Staphylococcus aureus possess a particular gene or some genes that encodes the expression, or they encodes the virulence nature of its, its genome, right, or itself as an organism to cause skin infection. I hope that is clear. Then we have UTI, and UTI is caused by what? We can drop that very quickly. UTI is caused by what? By what pathogenic organism? That's urinary tract infection. E. coli, exactly. E. coli. So E. coli, right, cause urinary tract infections. Now let me break your mind. Let me shock you, right? So presently at Genomark Up, we're embarking on a specific area of research that is focused on Staphylococcus aureus. So we are working with the entire whole genome sequence that we can. Hello? Hello, please. Can that person mute? Hello? Thank you so much. Thank you so much. Sorry, this is the first time we're using Microsoft Teams at Genomark. We've been using Zoom. So, okay. So let's proceed. Like I said, right? Like I said, we've been we're exploring the entire whole genome sequence of E. coli that we can get in Africa presently. And the idea that we have about that is that we want to explore the entire genomic structure such that we want to narrow down our research to specific genes of interest that are encoding the virulence nature of E. coli, right, in Africa. So we want to track it down. So this is one of the big research that we are doing currently at Genomic Up. We are working on over 6,000 whole genome sequence. Can you imagine that kind of work? Over 6,000 whole genome sequence. We're exploring it presently. And we actually have an idea of surveillance. Now, as we go on, you now begin to understand applications of microbial genomics in health. And one of it is surveillance. And that is one of the things we are doing currently at Genomic Up, such that we want to narrow down, like I said, to specific virulence structure, right, of virulence nature of, the E. coli that is causing urinary tract infections and other infections around it, all right, like diarrhea, right, and others. So let's proceed. Then another area of microbial genomics research is probiogenomics, okay, probiogenomics.
Sorry to disturb. Can you remove your chat slide so that your PowerPoint is visible, please? Thank you.
Oh, you can see my chat, right? Oh, sorry. Now, is it visible now? Hello?
Yes.
Yes.
Is it
Yes,
amazing. Thank you. Thank you.
That's
so,
very good.
Yeah, thank you. So an aspect of microbial genomics research is probiotic genomics. Okay. So, and this particular genomics research is applicable even in the industry, right? Especially in the food industry. So, and if you hear about probiotics, what's the first thing that comes to your mind? What's the first thing that comes to your mind when you hear about probiotics? Who is there? Hello? What's the first thing that comes to your mind when you hear about can somebody drop it at the chat room? The first thing that comes to your mind when you hear about probiotics.
Hello.
Hello, yeah.
Yeah, beneficial microorganisms.
Yeah, I understand. But all of you, exactly, I just got somebody. Every one of you guys have been academicians right now. So such that you are telling me the names of those probiotic bacteria. But when you hear about probiotic, I believe the first thing that should come to your mind is yogurt. Right? The first thing that should come to your mind is yogurt, or yeast, or those kind of probiotic food, right? So that's the first thing that should come to your mind when you hear about probiotics. Now, probiogenomics is an area of research, right? I hope the screen is clear now. So probiogenomics is an area of research that focused on the good bacteria. It's an area of microbial genomics research, right? That focus on what? On probiotic bacteria. And this area of research is applicable or is used even in the industry. Though pathogenomics is also used in the industry, such that you want to understand the virulence nature of bacteria that are affecting the food, production process in the industry. Some of these things I'm already saying it already, even though it's in the other slides, right? So that is an area of probiogenomics, such that, and in this area of genomics, you want to focus on the specific genes that code for the expressions of, the nutritional benefit or the health benefit of this probiotic bacteria. All right, because some of this, even in our body, we have some bacteria, right? Even in our mouth that are good. Now, we have some bacteria on our gut. They call it gut microbiome. Now, this gut microbiome are coding, right, for the benefits of for certain health benefits in human or in the host, including yeast. Exactly, yeast is also there, right? So that, probiogenomics is a very, very important area, very, very important area that is used, right? To do those that I've said. Now, another area of genomics research, right? Microbial genomics research is metagenomics. Metagenomics. Now, you see, metagenomics is used to explore both the pathogenomics and the probiogenomics, or both the pathogenic organism and the non-pathogenic organism. So metagenomics is the combination of both, such that in metagenomics study or research or area, microbial genomics research, you'll be able to have genetic information, right? You'll be able to have genetic information from both organisms that are probiotic, or those that are pathogenic, or those that are non-pathogenic, and those that are pathogenic. All right. So that is an area of microbial genomics research, such that it has been proven to be one of the powerful tools in the microbial field to explore, such that you have opportunity to focus on a wide range of community of microorganisms, and then you'll be able to study wide and have a detailed result at once from a specific environment, right? So it's like a very, very powerful tool, and we have different areas of metagenomics analysis as well, or metagenomic study, which we are going to definitely focus on. Okay, so let's proceed. Now, what are the applications to health? What are the applications of microbial genomics to health, right? So I've said some of them. One of it is outbreak detection and surveillance. Outbreak detection and surveillance. I believe, when COVID-19 broke out, right, one of the a powerful tool or cutting edge tool that was used, all right, to understand the nature of COVID, right, was through genomics, such that we're able to focus on understanding the nature, understanding the genomic structure, understanding specific genes and what those genes are coding for, right? Because there are certain genes that when you find, you'll be able to understand the behavior of COVID, such that you'll be able to understand the virulence behavior of COVID. And then one other area of microbial genomics research is in terms of variant analysis, right? Or variant study, or mutation study, right? Such that you'll be able to understand certain point mutations, so that it may probably it's coding for the pathogenic organism to be more virulent or less virulent, because some of these things are essential, right? They have their effect. Even when you talk about cancer, mutation in cancer. Mutation is what causes cancer. Mutation in specific genes, right? In human, right? That's what is causing cancer. So imagine if a virulence nature is mutating all over again. For instance, COVID-19 has been known to mutate, and to an extent where we had is it mega, or what was the last name of please, can somebody drop it at the chat room? What was it? Omicron. Thank you so much. Omicron. Thank you so much. Thank you so much. So for it to for COVID to evolve over time to such stage was because of the mutation, was because COVID, the SARS-CoV was mutating over time. It was mutating. It was continually mutating. And, some of the drugs that have been designed maybe at the earlier stage were no longer working because it has mutated, it has evolved, right? It has evolved. So, microbial genomics is a very, very powerful tool. Okay, microbial genomics is a very, very powerful tool, and it's used, like I said, in outbreak detection and surveillance. And this surveillance is one of the projects we are doing currently in Genomark Up. We want to build a database for specifically for for Africa, right, for some of these pathogenic organisms. So you are knowing now. So in case if there's any one of you who you are interested in the work, you can reach out. Probably if you are in Nigeria, you can reach out to us to join us at the lab. So we're doing it. We need more hands. So, and then the other aspect of microbial genomics is diagnosis, right? Diagnosis. So you can use microbial genomics to diagnose, right, this pathogenic nature of microbial organisms. Then the next one is precision medicine, right? Precision medicine. I believe you understand what precision medicine is. So you can actually explore, or apply microbial genomics when it comes to precision medicine. It's very, very an interesting area. Then we have drug discovery. Now, let me give you a gist about this drug discovery. What was the first antibiotics that was made? Who can tell us at the chat room?
Penicillin.
Amazing. And where do you think penicillin was gotten from?
Fungi.
Fungi.
Penicillin. Okay. So now this is just Now this is just to make you understand that, right? Understand that microbial genomics is a very, very interesting area, such that through microbial genomics, you can actually discover new or novel drugs, right? Novel medicines from it, all right? For the benefit of mankind, to treat infections. You can discover new antibiotics. Now, one of the area of drug discovery that deals with microbial genomics is what we call therapeutic peptide. They are what we call antimicrobial peptide. Now, this therapeutic peptide, they have, it has been shown to be a very, very important area to explore, to discover new antibiotics to deal with infections that are caused by pathogenic microbes, right? So that is a very, you can make research on that. What is therapeutic peptide, right? Or what is therapeutic peptide research? We are doing that currently in at Genoma Corp as well. So such that we discovered a novel peptide from a particular microbe that can actually combat skin infection caused by Staphylococcus aureus. And then we are doing that research and development currently, though we're at the in silico level, but we are hoping that very soon we'll graduate to the wet lab experiment, such that we'll be able to validate the peptide, because we have a lot of peptides. So we have to validate which one is more potent, and then we do some animal trials. So can you begin to see the area of microbial genomics? That is a very, very powerful area, such that if you have an interest in genomics, one of the aspects of genomics that you can explore is microbes. So if you can actually focus on human genomic structure, but another area, like I said, is what the area of microbes. And another area we have is nutrition and diet. And in terms of nutrition is when you're exploring the probiotics organism, or when you are exploring the you're exploring the genomic structure of pathogenic organisms in nutrition, right? Because some organisms causes food decay, and some cause food poisoning, right? Food poisoning. And for you to do that, you have to be able to understand the genetic structure, the genetic composition of these pathogenic organisms that are causing food poisoning, or that are causing food decay in those specific food that you're focusing on, right? So we have an applications to agriculture. But before I proceed, do we have any question? Before I proceed, do we have any question?
Please go.
Okay. So if there's no question, we're going to proceed. So if you have a question, you can just drop it at the chat room. Okay. Thank you so much. So, as I was saying, let's proceed to applications to agriculture. So, applications to agriculture, we have investigate plant infections. Okay. You can actually focus, or you can actually explore microbial genomics in terms of applications to agriculture, right? Based on what? Investigating plant infection. Okay, so we have a lot of plant infections that are even caused by bacteria itself, right? That are caused by bacteria itself. So one of our collaborators, right? I believe probably she's online right now. She's from Belgium, right? So we were doing a particular research on a plant, and we were exploring a particular pathogenic bacteria. And we get to understand that this pathogenic bacteria has developed a lot of a virulence nature because of the genes, right, or the putative function of functional proteins, right, that are coding for the expression of that specific nature. So these microorganisms are affecting those plants. In fact, not just in Africa alone, even in other geographical areas. All right. So it's really a very interesting area to explore, such that when you get this bacteria, or when you get these microbes that is causing plant infections, you'll be able to streamline, like I've been saying since morning, you'll be able to streamline to those specific genes, understand them, understand the mutation. And then one interesting thing about this is that when you explore this pathogenic organism in using genomics, you'll be able to discover what we call drug targets. All right? You'll be able to discover what we call drug target. Now, when you discover this drug target, that is like giving you a solid foundation to begin a drug discovery research, right? Because a drug target of a particular pathogenic organism is a potential target to be able to, such that you'll be able to deal with that specific infection, or will you be able to deal with the infection that is caused by that specific bacteria? Because this drug target, it shows that when you target that particular site, or when you target that particular gene, right, you'll be able to probably inhibit or kill the pathogenic bacteria in question. Now the other area is investigate soil and plant nutrition. Investigate soil and plant nutrition. So, if you're talking about agriculture, soil is very, very important, right? It's a very major pillar when it comes to agriculture. Now, you got to discover that in agriculture, when you plant some, when you did some plantation, right? When you did some plantation, when it comes to plant, right? Because there's animal side of it as well. So when it comes to plant, you did some cultivation of a particular plant, but you get to discover that this plant is dying. Like all the plants, mostly you're expecting 100% yield and you're getting maybe 20% yield, right? Now, the reason is because probably there's some microorganisms in that land that are causing that are causing that particular issue, right? That particular case. So you have to investigate that soil. And in investigating the soil, one of it is that you want to know what is the composition of microorganisms in that soil. Now, are there microorganisms that are, virulent, that are virulent, all right, that are pathogenic to that particular plant in question? Because you may plant another, you may cultivate another plant on that particular soil and the plant survive. It means that there is a specific microbes that is pathogenic to the plant that does not yield well, right? The plant in question, the plant that is not producing well, right? Or the plant that is dying on that particular soil. So when you get the soil sample, right, you take it to the lab, you do some culturing, and then you take it to the sequencing lab, which some of the things we are going to explore as well. And by the time you get there, you'll be able, you move next to doing some kind of analysis. Now, doing that analysis, you'll be able to know the composition of the bacteria, and then you'll be able to streamline your study to based on your research to so I have this, there's this particular microorganism in the soil, and at such the plants cannot survive there. So what you need to do is probably maybe get a particular chemical that can actually kill that particular microorganisms in that particular soil, right? Or you did some other research in order to deal with it. And then other research you can do is that by the time you begin to explore that particular bacteria in pathogenic bacteria or microbes in question, you'll be able to, okay, I want to develop or innovatively discover or develop a particular solution that can deal with that. If there's no solution for that, right? Maybe probably the microorganism you saw is novel, right? So you have to discover to create a novel solution for it. And that is a great idea, right? Innovative idea in terms of when it comes to micro, because you see, the genomics industry right now is growing. It's really, really growing. So the next is processing of agricultural products. I've already talked about this, right? When it comes to industry, when it comes to the industrial aspect of microbial genomics. Okay. I mean industrial aspect of food. Okay. So the next thing we have in here is in agriculture we have animals, right? We have in terms of livestock as well, right? So. And that a lot of livestock are dying as well, right? It could be based on pandemic or whatever it is, right? So investigating the microorganisms is also very, very important, okay, to deal with such case in terms of livestock agriculture. Then we have NGS data metagenomics, okay? Now, we've done with the research and the area of research and applications of microbial genomics. Now, in the previous slide, we talked about metagenomics, right? As one of the research area, right? One of the research area. And we talked about that this metagenomics combines both probiogenomics or pathogenomics, right? We can use metagenomics to study gut microbiome. We can use pathogenomics to study the microorganisms that are found in yeast, right? When it comes to yeast, you do that. And then, or you can use metagenomics to do a pathogenomic study. And then it means that metagenomics is applicable to health, and it's also applicable to agriculture. And then as we go on, you now begin to have a clearer picture of this aspect of research. Then we have NGS data metagenomic study. Okay, like I said, NGS data metagenomic study. Now, I already told you what metagenomics is, but we are going to define it as we go. So, but what is NGS here? What is NGS? Please, can somebody drop it on the chat room very quickly? What do you think is NGS? At the chat room,
Next generation sequencing.
Oh, okay, thank you. Okay, thank you. Next generational sequencing, okay? And I believe you guys, you all understand what sequencing is all about. So, but when it comes to sequencing, like the process of generating genetic data, right? Genetic material from a specific life and of a specific organism, right? It can either be microorganisms, plants, or animal. So that, but we have what we call next generational sequencing. We have what we call Sanger, right? There's Sanger sequencing. And then we have next gen sequencing, which is next generational sequencing. Now, you see, next generational sequencing is kind of unique and powerful, such that it's focused on a wide range of genetic covering, right? Of genetic makeup of an organism. So that's next generational sequencing data metagenomic study. So it means that you use next gens to do metagenomic study. Now, what is metagenomics? Okay. Though we have the practical applications, like I said, so it's to study a specific community of microorganisms. It's known for its application in health and agriculture, such as pathogen and plant disease identification, disease-resistant breeding, right? Plant pest control, weed management, abiotic stress management, post-harvest management, discovery agriculture. And do you even know that metagenomics is also used in the oil industry? Do you know that? If you know, can you drop a yes at the chat room? Metagenomics is also used in the oil industry to investigate the microbes that are found on this sites, oil sites, right? So I don't think anybody knows. And you know, this area of metagenomics, when it comes to oil industry, most times they do work on metatranscriptomics. So it's combination of metagenomics and transcriptomics together. So, and then we have shed light in the what? Development of probiotics and so much more, right? I've already said it. So let's proceed. Metagenomics. Now, metagenomics is also called environmental genomics or ecogenomics or community genomics. It's the study of genetic material recovered directly from environmental samples, such as soil, water, feces, clinical samples. It could be, when it comes to water, it could be pond, it could be fresh water, it could be lake water, right? It could be any kind of water. And when it comes to soil, it could be any kind of soil. When it comes to feces, any kind of feces from any kind of animal. It could be man, it could be poultry, it could be cow, it could be any kind of animal. Then we have the clinical, right? So, you know, when it comes to feces, for example, when probably a pandemic or when a particular infections, maybe diarrhea or cholera break out in a particular community, right? So they gather the sample and they could use metagenomics to exploit, right? Such that they'll be able to have a wide range of study to study the community of microorganisms. So metagenomics focus on what? The community of what? Of microorganisms that are found in a specific area, in a specific site. And imagine if you are doing metagenomic study on a particular soil, and then you get the culturing, you do the sequencing. Now, in that particular genetic material, you find genetic material that belongs to either probiotics, that belongs to bacteria. Maybe you find most to be bacteria. It depends on how you streamline your culturing, right? Other one is fungi. Another one is virus. In fact, you could even find some human samples in it, depending on which site you're actually bringing it from, you're getting it from, and how you streamline your culturing at the lab. Then we have the approach include the target sequencing and shortcut sequencing. Now, the target sequencing is focused on amplicons and specific genes useful in targeting a large diversity of organisms. A large diversity of organisms. Remember that. You know, pathogenomics, right? Pathogenomics focus on the entire genomic structure of a specific organism, maybe E. coli. But you see, in metagenomics, you find different strains of E. coli in it. In fact, you find different strains of maybe Staphylococcus aureus. You find different strains of, different kinds of, even not just bacteria, even, virus as well. So, and then we have this target sequencing is focused on 16S RNA, while the shotgun sequence is focused on whole genome shotgun, WGS metagenomics, whole genome shotgun, right? Not whole genome sequencing, right? Whole genome shotguns, right? All shotgun sequencing. I believe I'm right, right? Then it allows a comprehensive sampling of all genes in all organisms present in a given complex sample. It allows a comprehensive sampling of all genomes in all organisms present in a given complex sample. I hope that is clear, right? Because you see, the target sequencing is focused on diversity. It's focused on classification, understanding what area of diversity did they found, beta diversity, alpha diversity, right? So you'll be able to understand them. You'll be able to, because in diversity, you're exploring the taxonomy rank. You are exploring what is the abundance, what is the relative abundance of, of bacteria or species, right? Or specific strain belonging to a species. You'll be able to narrow it down. When it comes to structural sequencing, it can also do biodiversity, right? It can also do taxonomy classification. You get to understand as you proceed. Then we have the principle, right? This is the approach, and the principle. It relies on the genomics analysis of sample from a complex environment containing more than one organism, hence providing a visual composition of this sample. A visual. You'll be able to know. You'll be able to know the name of specific strains, specific organisms, and you know the abundance in that particular environment, be it soil, be it water, or So I hope it's understandable. Please, can you drop a chat room? Is it clear? With a yes? With a yes. Hello? Is it clear?
It's clear. It's clear. Yeah.
Amazing. If you want the PPT, okay, we'll look into that. Okay. Thank you so much. Thank you.
Thank you.
Okay,
we
so
really
let's
appreciate.
all right. So let's proceed. Let's proceed. So we have the principle. Okay. So I've explained this. So we have the process now. As a PhD holder, right? Or probably you want to do your PhD and you're cracking your head. You want to understand what area of research you want to engage in. You want to focus on, right? So the process, this is the process, right? You first you embark on sample collection. You go to the site, you collect the sample. When it comes to metagenomics, but first remember you have to know what you want to do. Is it 16S metagenomics or is it shotgun sequencing? Is it target sequencing or is it shotgun sequencing? So if it's target sequencing, you want to do biodiversity. You can download similar papers on 16S metagenomic study and then see how it is, right? And then the other one is shotgun sequencing, right? So but this is the process for both, right? You get the sample collection, DNA extraction, right? That's at the sequencing lab, right? Then we have the sample preparation, still sequencing, PCR stuff, right? Then we have the sample analysis. Now, all these are huge work because sample collection, you go to the site, you must know the site you want to get it from, and why you are getting it from that site. You must have a research objective. Then we have the DNA extraction, right? DNA extraction. You have to extract it. Sample preparation, sample analysis. Now, we are going to be looking into sample analysis. All right, we'll be looking into the sample analysis. So let's proceed. Now, the first step involving analysis of metagenomics WGS sample, right? Metagenomics old genome shotgun, right? Old shotgun sequencing sample. Then we have assessing metagenomic sample on NCBI database. Now, these are the steps involved. So assessing metagenomic samples on NCBI database, that is if you don't have your sample. But if you have your sample, you have to skip this and go to the next. The next is introducing, so we are going to introduce you to a pipeline for metagenomics analysis. Okay, so you must use a pipeline, because we have different pipelines for metagenomics analysis. So which pipeline do you want to use? Okay, to do your analysis, you must sit down to do a visibility study. Okay, then we have the raw rich processing and genome assembly. Because when you have this sample, It's raw. It's raw, right? So there are areas that there are some errors, so you have to trim them out, and then you have to assemble. Then we have the taxonomic classification and the evaluations of results, right? So taxonomic classification here talks about the diversity, what taxonomic rank specific organism belongs to, right? I'm going to show you a visualized chart, taxonomic chart, to show you how it looks like. Then the comprehensive metagenomics analysis. Comprehensive metagenomics analysis covers both the annotation, the prediction, the gene prediction, right? So because, don't forget, in the taxonomic classification, you will know organisms that are pathogenic and not pathogenic. You'll be able to streamline to a specific organism of interest that you're looking for, that is specific in your research. Then the next thing is comprehensive metagenomics analysis that talks about the annotation, when you focus on the gene. Remember, this is WGS, not 16S, right? This is shotgun. This is shotgun sequencing, okay? This is metagenomics shotgun sequencing, okay? And when it comes to shotgun sequencing, right, we already understand that it focuses on the genes of the organisms that are present in the environment. So it's wide. It's beyond just individual sample, right? Right?
Sorry to disturb you.
Okay.
Just to understand something here, whole genome sequencing and shotgun sequencing. And
no, not whole genome. No, we're not talking about whole genome. Whole genome is specific. It's specific to an individual organism.
What is WGS?
Now, this is whole shotgun. Whole shotgun.
Okay.
We're talking about shotgun sequencing.
Okay, you are not speaking about whole genome sequencing, right?
No. We're talking about shotgun.
Okay.
This talk about allowing the comprehensive sampling of all genes. Now, if you're talking about all genes, you're talking about whole genome, isn't it? The whole genome of the organs that are present. But this time around, it is shotgun sequencing, because if you're talking about metagenomics, it is shotgun, right? Is that clear?
Okay. Thank you.
Yeah.
Thank you.
Then we have summarizing comprehensive results. Then we have evaluating the antimicrobial resistant genes. We have evaluating the virulence factors. We have the mobile gene elements. And then we have the computing and visualization of results. Now, these are the steps that are involved. Okay?
Mira lo que me escribió. Thank you for your email. We will discuss after the workshop. ¿Para que me va a escribir después de la presentación? Sí, ¿verdad?
Please, can you mute? Please, can you mute? Sorry. Hello?
Thank you for your email. We will discuss after the workshop, see?
Please, who is speaking here? Is it Josh? Andres? Andres, please, can you mute? Andres. Thank you.
So sorry.
We have assessing real life data samples from NGS. So this is just when you want to get the samples. All right, then we have introduction to the pipeline database. So I would like to tell you right now that these are the steps, right, that are involved in exploring the metagenomic sample, such that you'll be able to focus, or you'll be able to know, you'll be able to do a wide range of study, right, when it comes to a specific sample, when it comes to a specific metagenomic sample, probably soil, maybe water, or anything you want to focus on. Now, these are the steps involved. Now, these steps involved, we will be having a practical session, okay? There will be a practical workshop, a hands-on practical training, which we are going to be introducing you to, okay? So, and in this practical training, we will cover all these steps. So we'll be working with real-life data from NCBI database. We'll show you we'll show you specific pipeline tools, right, where you'll be able to do metagenomic analysis, be it taxonomic classification. So we're going to proceed from raw-rich processing and genome assembly. Then we're going to focus on taxonomic classification and evaluations of results, show you how you can evaluate your result, how you can interpret your result. Then this is a chart of taxonomic classification. We call it Kraken, right? Can you see here we have bacteria, right? We have the root, we have the eukaryote, we have the Homo sapiens, and as you go, so we have all. So I think there are other ones, but by the time you begin to explore different metagenomic samples, you'll be able to see those who has virus, those with no heat, those that have bacteria, and these are the species. And then you'll be able to understand how you can proceed to, from the phylum down to, the species, right? And we have comprehensive genome analysis, where we'll be doing genome annotations and you'll be predicting AMR genes, prediction, virulence factors, mobile gene elements, transporter genes, drug targets, and all those things we'll be doing them. So we have evaluating the antimicrobial resistant genes. But if you ask me, what is antimicrobial resistance? This is the ability of bacteria to resist the effect of antibiotic drugs that are commonly used to treat them. That is antimicrobial resistance. And resistance arises either through natural resistance certain types of bacteria, genetic mutation, acquired resistance from another. Now, in acquired resistance from another, this is where mobile gene element is associated with, right? Or associated to. Acquiring resistance from what? From another. Because to acquire resistance from another, there must be a mechanism of what? Of antimicrobial, of mobile gene element. So you understand what mobile gene element is as we go. And one of the other things you'll be looking into is AMR genes, what antibiotics they're resistant to, and also we're going to explore the resistant mechanisms. Now, what's resistant mechanism? This is the mechanism that these antibiotics are using, right, to resist those bacteria. All right? Then we have evaluating virulence factors and genes. And when we talk about virulence factor, we're talking about the pathogenicity determinants, how these bacteria are pathogenic. Now, when we're exploring the shotgun sequencing metagenomic sample, all right, we are going to begin to see in the analysis, right, you see all the genes that are contributing to the virulence nature of different bacteria, because the names of those bacteria will also be there, right? Then we have facilitated the successful colonization and survival of the bacteria or cause damage to the host. Now, this is the definition of virulence factors or virulence genes, right? They facilitate the successful colonization and survival of the bacteria or cause damage to the host. Either that is either specific to the plant or specific to host, to human, right? Specific to humans. So we're able to explore this. Then we have mobile gene element, like I said, segment of DNA that encode enzymes and other proteins that enable movement of genes from within genomes or from one genome to another. That means one bacteria to another, one from different strains of the same species. Mobile gene element. And this is how resistant genes of virulence nature or virulence genes spreads. And we currently have a mobile, a global threat presently on AMR. How many of you know about that? Can you reply with a yes? If you know about AMR, WHO have been talking about that, and then we just previously did the AMR awareness week recently, talking about the present pandemic that is going on around in the world, right? It's like a pandemic. It's a global threat. AMR resistance. Anybody understand this or knows about this? Can you drop a yes? Okay, so let's proceed. So then we have mobile gene elements, and then some other aspect where we have visualization of results, where you'll be able to drop heat maps, right? Where you'll be able to drop, drop some charts and things like that. So looking at the practical classes, it is 100% practical. Now, I would like to tell you that the practical class is going to start tomorrow. Okay? The practical class is going to start tomorrow, where you will learn how to retrieve real-life data from NTBI database. Now, I did a particular research with someone who was doing his PhD in China, and he worked on data from NTBI database to do his research. Though he did a very comprehensive study, right? Did a very, very comprehensive study. But what he worked on was WGS, where he explored the biodiversity. He explored the virulence genes. He explored the AMR. He explored the mobile gene element. He did some network analysis. Now, we even have nano sequencing samples as a platform for shotgun sequencing of metagenomic sample. And there are, some analytical process like that, okay, that you can embark on. Still the same kind of process or steps, all right? So in this class, we're going to show you, in the practical classes, which is the workshop, right? We're going to show you how you can do this analysis, right? How you can do this analysis, and from beginning to the step, from the very basic to the advanced step. And like I said, the practical class is going to commence tomorrow, okay? And the actual price of the practical class is something you can actually afford, okay? So, and we're going to give you access to the videos, okay, where you know how you understand these things step by step, get the samples from NCBI database, and do all the steps that I've mentioned earlier, and then how you can visualize your results, and then do your evaluate your work for a complete research paper, okay, for a complete research paper. So let me quickly show you. You can just go to www.genomarkup.com/metagenomics2, okay? www.genomarkup.com/metagenomics2. So the price is actually, can you see? This is understanding metagenomics. You see one of our research papers that we did. You can read the research paper, all right? So we have, if you want to embark on research, life science research and students, medical practitioners. So this is it's for you. Now, I just want to show you the price. The price is actually $100, but we are releasing it now at $20. Okay, we are releasing out at $20. Give me a minute. And we have bonuses for you as well. Okay, because we understand that. Yeah, so this is it. So as you can see here, we have the modules, okay? We have the bonuses, we have the modules, and then we have, okay, let me just explore the modules so that you can see. So you get access to the modules and then live sessions on Zoom, and it's starting tomorrow. So we're going to explore all the steps that we have shown you. And this is the price, okay? It's previously $100, but we're releasing it at just $20. Just $20. For those that will register now, we also have bonuses for you. And the bonus you have is access to paid activation key with lifetime access to Genius software. Now, you see, this Genius software can do a lot of analysis from phylogenetics, transcriptomics, variant calling, comparative genomics, map reading. So we're going to give you activation key. This activation key currently costs, I think, more than $400. We're going to give you an activation key for life, lifetime activation key for this Genius software. It's a genetic software, it's a genomic software for doing different kinds of analysis. Okay? Then we have slot for 6 months mentorship on bioinformatics related topics. So it means that you have slot for 6 month mentorship to discuss with one of our experts, probably on your projects, right? Maybe some challenges you have, maybe you have some questions, and for those that actually enrolled for the course, all right, between now and tomorrow, you get access to these bonuses. Then we have the third bonus is that we give you lifetime access to one of our online bioinformatics course, with practicals such as whole genome sequence analysis. This is whole genome sequence now, not metagenomics. With this whole genome sequence analysis, you'll be able to explore pathogenic or probiotic genomics, okay? Then multiple sequence analysis. And then we have using Genius software, okay? And some genomic pipeline too. Then we give you access to a resource package of over 50 bioinformatics tools and pipeline to perform various kinds of bioinformatics analysis and interpretation of results, right? So because we have different kind of bioinformatics tools and pipeline, then we give you that package. The package is available. So such that you have, different tools for doing different kind of genomics analysis. All those, the links will be there. It's a package, actually. And then you get it. And these are the words. But these are just bonuses for you. So it means you have two opportunities here. To register at $20 and get access to a bonus that is worth $1,000. Okay? Don't forget, if you're a PhD student, with this course, you'll be able to have access to one of our, expert, so that you'll be able, you will discuss your project, okay? You discuss your project topic. If maybe you don't know any project topic, you'll be able to discuss your project topic with that expert. We'll fix a time where you book to be able to attend to this. So, in case you are interested to register, okay? In case you are interested to register, you can go to this link. I'm about to share the link right now. The link. So that's www.genomarkup.com/metagenomics2. So you can just go there and register, okay? Or you reach out to us online on WhatsApp. Reach out to us on WhatsApp on +2349077191794. You can reach out to us so that you'll be able to register for the course. All right? So any question? I'm going to take question right now. Any question? Any question? We're taking questions right now. Don't forget, between now, because we already have people who have registered, so we have limited seats, okay? So if you want to get access to this course and you want to learn how to do the analysis, okay? So just, okay, thank you, Abidem Abubakar. Thank you so much for sharing the link. So I'm going to take question right now, okay? No, workshop is online. We're going to give you 100% practical videos, okay? We're going to give you 100% practical videos. 100% practical videos. Okay? So you do that for 3 days. And at the third day, we're going to meet on Zoom to clear your doubts. Okay? So you have 3 days to go through the modules. We're going to give you the videos. You go through the videos for 3 days. And please, there's no lifetime access. We'll give you the videos for maybe some weeks, maybe 2 weeks or a week more. Okay? So after the 3 days, we'll give you more days or more time to be able to study them, to be able to understand the videos. Okay? So, and then we give you certificates. There'll be certificates of completion. Okay? There'll be certificates of completion. And if you're a PhD holder, you can enroll for this workshop. And if you are confused about your project topic, you have one of the bonus is that you have 6-month mentorship. Like I said, 6-month mentorship for your, to ask your project. To ask some questions if you have challenges, right? If you probably you want to discuss your project with us, it's a slot. It's a free slot, so you book it, but only if you enroll for the workshop. Okay? Only if you enroll for the workshop. So if you are interested for the workshop, please quickly register or contact us. Okay? So we're going to open the WhatsApp, the group link. So in case you are interested, you can just signify at the group link, or you chat up the group admin. Okay? You can chat up the group admin. So I want to take questions. Please ask your questions. What time is the workshop? Like I said, it's online, so it starts from tomorrow. So you have 3 days to go through the video modules. So every day we give you access to the first day modules, then second day modules, and third day modules. Okay, and all those steps are there. And then we give you extra days to go through it again so that you'll be able to have full understanding. Then we're going to give you certificates of completion, okay? Certificate for the workshop. And then you have access to the bonuses, okay? You have access to all the bonuses, like I said. So I think there's a question here. I'm a student in, master's, you're doing your master's, molecular biology and particularly at Pan Africa University in Kenya. I did the registration already. Really want to discuss research topic with you. Okay, if you did the registration, please, before you can get access to the bonus to discuss your research project with an expert at Genomarkup, right? You have to enroll for the workshop because it's part of the bonus, okay? That's what I said. Then we'll be able to discuss your project topic, or we'll be able to discuss your project and know how we can help you in the course of the 6-month mentorship, okay? Then we have a biotechnology student sequencing my culture from cornea female sample. We isolate fusarium, okay? From your experience, what kind of analysis or statistical analysis we can work with our genome result. Okay, good. George Andres Ramirez, please can you contact me on WhatsApp if you're on WhatsApp. If you're not on WhatsApp, contact me on at gmail.com, genomarkup@gmail.com. Contact me on through the email so that we can discuss. Okay, because looking at it, definitely we can actually come up with a workflow, okay, for your research, and then such that workflow will be able to produce high impact factor journals for you. Okay, because if you enroll for the workshop, these are some of the opportunities you are going to get. Okay, so that we'll be able to discuss your project, and then your project will be able to, produce a high impact factor journal. Genome Club has been able to work with a lot of clients across the globe, from Canada, America, China, and the rest. Okay, so you can discuss it. Okay, I believe you are still here, George Andres Ramirez Socorro. Please,
your email again, please.
Are you there? Hello? Hello? Can you hear me?
Your email address, please.
Okay, genomarkup@gmail.com. I just sent it. Check the chat room. genomarkup@gmail.com. genomarkup@gmail.com. Yeah. So just chat me up so that we can discuss your project. Okay? Chat me up so that we can discuss your project. Please, I need questions. You can ask your questions. I'm taking questions now. So
okay.
Somebody said tomorrow with the first module, you will give all instructions for the practice class. Exactly. Yeah. There'll be all instructions, how you get access to the modules, to the video sessions. Is 100% practical videos. Step by step, you will become an advanced microbial analyst. Okay, advanced microbial analyst, such that you'll be able to understand, you'll be able to do this analysis whenever you get the real life data. We'll show you how you get the real life data. All right, and then I will show you practical step by step approach to to analyze your sample. Okay, step by step approach. So yeah, your question.
Hello, thank you, Mr. Oluwaseyi. Can you hear me, please?
Yeah, go on.
Okay, so actually, I think we chatted there some times ago concerning the programming language that is going to be useful for the metagenomics analysis, and you mentioned Python, and I want to ask, what do you think? Like, what is the level of proficiency that is required in order to be able to perform well in this workshop?
Okay, like I said, I think if you go through I think you are still seeing my screen, right? So it said learn to practically explore NGS metagenomics analysis and metagenomic sample, right? Analyze and comprehensively or comparatively interpret, because you cannot just analyze one sample. You must be able to have a detailed understanding of how you can comparatively interpret it. Interpret the results in terms of visualization and computing of data in 4 days without any prior knowledge of bioinformatics. Okay? Without any prior knowledge of bioinformatics. And mind you, we're exploring, in this workshop, we're exploring the WGS, okay? Okay, well, the shotgun sequencing, okay, for soil, water, whatever you want to use, okay? So, without any prior knowledge, either you understand Python, you don't understand Python. Either you have Linux or you have Windows platform. The workshop is for you. It is a step-by-step guide, okay? It's a step-by-step guide. But as a scientist, you want to explore the area of bioinformatics, right? So if you want to do that, you also need Python. You also need the knowledge of Python. It's very, very important, because there's some algorithms, or there's some pipelines on GitHub that you can use to do quite a number of different kinds of analysis. But mind you, the era of bioinformatics has moved beyond the scripting, the writing of the scripts, and running those scripts for analysis. One of the area of bioinformatics that is coming up right now is being able to run your analysis, on already built pipelines, databases. All right, already built pipeline databases. So that's an area of bioinformatics as well. But the thing is, information is lacking. So the more information you have, the more open, the more information you have, the more you are open to do, different kinds of analysis. The more, bioinformatics analysis becomes easier for you. The more analysis of samples of genetic data becomes easier for you, because that is information for you. That is information. So information is very, very important. And in this workshop, we're not only giving you information, we are also giving you practical experience. It's on-us training, okay? Practical experience. So, any question more? Thank you for that question. Okay, thank you for the very informative meeting. I am graduate student in biotechnology and specialty in biosecurity and bio defense. I look forward to work with you. Okay, thank you. I believe you're registered already. Thank you, see you. Okay. What's the requirement of laptop? Like I said, well, because we're going to be giving you Genius software, I think you need a minimum of maybe 2 gig RAM, okay, 2 gig RAM, and a minimum of maybe 80 ROM, right? 80 gig ROM, okay? You need a minimum of that to be able to install the Genius software. So the Genius software runs, on different kinds of bioinformatics analysis, and it's lifetime access. So it will work. So what's the requirement for? Okay, I think we've done that. Can I get the contact of doctor from DRC among us here? Sorry, is there any programs to use? Okay. Any more questions? I don't know if we have any more questions previously.
Hello, sir.
Yes.
The training, is this self practice, or there is going to be a live section?
So it's both. It's both. Okay. Step practice such that we are giving you the videos, practical videos. Okay, step by step. Are you there?
Yes, I'm listening.
And then we have live sessions as well. Live sessions.
Live session. What are the sessions? When is it starting? And I mean the time.
You have 3 days to go through the modules. Then on the last day, we have live session. Okay, to clear all answers. We have the live session like this to clear all your questions, to show you how to do your bonuses, how to get access to your bonuses. All right, how to do some other kind of analysis through the bonuses as well. And then we clear all your doubts. If you have questions from the modules, we clear them. So it means that if you are busy, if you're a very busy person, you have the modules on your free time to go through them at any time, even in the midnight. Okay, to study it. And then after these 3 days, we still give you more days to be able to go through to study them again. Okay.
Okay.
Hello, sir.
Yeah, go on.
Hello. Okay. You said the video, they are not gonna be lifetime video. That maybe after a week or 2 weeks, they won't have access to them anymore.
Yes, but then listen, that is why you have the mentorship session for 6 months, okay, as bonus, such that if you encounter any issue along the line, you just need to reach out to us. Probably give you access to it again, okay?
Okay. But sir, you also have to understand the fact that some of us, though we've not even started our bench work already. Maybe it's even in a year time or after 6 months. So like, I don't know if you can just try to extend our access to the video, because most time it's really good for us to have access to the video. So if we are confused, then we can quickly run back to the video and get the clarification.
I understand. We'll sort that out. We'll look for a way around it. Okay. We'll look for.
All right. Yeah.
Yeah, I understand. I understand what you're saying, and it's very crucial. Okay, it's very, very important. So definitely we'll
all right. Thank you for that.
Yeah, thank you. Okay. So, any more questions? So, any requirement for lifetime video? Okay, I think we'll talk about that. So, amazing. Thank you so much. Dr. Adejari. Thank you. Thank you. Okay, so I desire questions. So you guys can ask your questions. You can ask your question as quickly as possible. Your questions. So I'll be dropping the link for the registration again. So just register and then we'll get access to you, or reach out to us on WhatsApp. Okay, 771917. Reach out to us. Next one. Reach out to us on WhatsApp and then we'll sort it out. Okay. All right. All right. So if there's no question, we have just a few minutes to round up. We have a very few minutes to round up. So this is an opportunity for you to ask your question because we are rounding up already. Okay. So please.
I have a question, please.
Okay, go on. Yeah, go on. Go on. Go on. I'm with you.
Okay. Actually, students in Kenya, Pan-African University, in molecular biology, biotechnology. And we are just about to start the research because we are almost done with our coursework. And I'm yet to decide about what we'll actually do as research. But at least I have some ideas. I was thinking of maybe trying to screen lactic acid bacteria from fermented food in DRC and try to find out those who are able to play the role of probiotic, and at the same time they can be able also to produce amylase for the fermentation of cereal-based food. So for the identification of those bacteria, I'm planning to do the 16S sequencing, but now I think from the session of today, I really want to do something more exacting, like the use the next generation sequencing for the shotgun sequencing, to discover the microbial community from those fermented food, maybe come up with more information that's than what I could have maybe from just the 16 gene sequencing. So I would like to know whether it's really affordable for a master's student to use those actually cutting edge technology like next generation sequencing, or to do what the whole genome sequencing. Is it really affordable for a master students, or maybe it could be also time consuming because we are given just 1 year to do research, so I don't know, will it
1
be
year?
really
Did you say 1 year?
Yeah, 1 year just for research.
Okay. Have you started already or not?
Not yet. We are to start.
You are doing your visibility study, right?
Yes, exactly.
Okay, good. Now, you see, I believe you can do metagenomics. I believe it. Okay.
Yeah.
And yeah, and if you, I know you have, like you said from your explanation, you have probiotics in mind, right? Probably gut microbiome in mind,
exactly.
or yeast in mind, right?
Yeah.
So I would try to advise you. You have to make your decision. Okay, is that you focus on probiotic as an individual organism, okay, or you go with metagenomics.
Okay.
Now, if you are going with individual organisms, it might maybe cost more, because you see, shotgun metagenomics is not like as cost as before. Okay, it's not like costly like before. Okay, so if you go with shotgun sequencing and you get one you get a sample and you get a read of metagenomic sample from whatever environment you are bringing it from. Okay, you'll be able to do a comparative study with probably other genomes within Africa or other genomes across Africa.
Okay.
Do you understand? But what I'm just trying to tell you is that both of them are still visible, but you just have to make your decision. Do you understand? Both of them are visible. If you want to go with probiotic as an individual organism, like the organism you mentioned, it's still okay. You can still do comparative study so that you'll be able to focus on specific genes of interest. But if you are going with metagenomics, it's still okay. But you should do shortcut sequence if you want to still focus on some of those genes. Okay?
Yeah. Thank you.
Is that clear? Yeah. Hello? Is there anybody with questions? I think there's some questions here. I'm a graduate of biochemistry. I want to study biotechnology. Is relevant to this microbial genomics? Yes, biotechnology is relevant to microbial genomics, definitely. So if you want to study biotechnology, there is an area of genetics. Okay, there's an area of genetics, and one of the area of biotechnology in genetics is called genetic editing. Okay, it's called genetic editing. It's also in microbial genomics. But you know, you cannot begin to explore microbial genomics in genetic editing without understanding the microbes itself. So you use microbial genomics to understand the nature of a microbe, and then it is from there you now begin to deduce the biotechnology aspect of it. We deal with CRISPR, all right? We deal with CRISPR, and CRISPR is genetic editing. I don't know if Yusuf Sadisu is still here. Yusuf Sadisu Ibrahim. If you are still here, I'm answering your question. Okay, it's relevant to this. Yeah. So you can understand, you can learn CRISPR. CRISPR is an aspect of biotechnology. Okay, where you do genetic editing. And one aspect of biotechnology is also probiotics, where you do probiotics engineering. Okay, there's engineering in probiotics. Okay, and then before you can be able to do that, you must understand microbial genomics. There's an aspect of biotechnology even in animal where you be able to edit some gene, but then you still need to understand genomics. But then I'm not sure if when it comes to animal, well, see, this thing is intertwined. Okay, one thing depends on other. Okay, you cannot single-handedly study one area and forget the other, right? So that is why genomics is genomics. Okay, I hope I'm able to answer your question.
Yes, thank you.
Okay, then yes, I'm a student. Masters in biotechnology, genomics, and bioinformatics is really important. Okay, thank you. This may not be related to metagenomics, but I would like to know how one can determine the size of PCR product expected from the sequence of the primers. Okay, there's the size. Well, you see, that is why in this metagenomics we also talk about rich processing and genome assembly. Now, in rich processing, you'll be able to understand the size of the raw read data. Now, it's after when you assemble that you now know the actual size, because assemble is about reconstructing the entire raw data into the original sequence. Because in the raw data, you have fragmented sequences that are scattered abroad, belonging to the same conserved region. So it is in assembly that you'll be able to assemble them and rearrange them to the same original sequence space, right? Or the position of the conserved region of those fragmented sequences will be rearranged and then will be produced, where you now have the original size of the entire raw data sequence that they were sequenced or produced in the lab through PCR process. Bontlu Ngazimbi, please, did I answer your question? Are you still here? Ngazimbi.
Yes, sir.
Okay. Yes, sir. Okay, you are here. All right. I'm actually an undergraduate student planning on using targeted sequencing for my final research project, although I have no experience in bioinformatics. Okay, and I believe that this particular metagenomics class, like we said, we'll be able to give you a heads up. Okay, we'll be able to build your foundation to be more solid, and then you have understanding about genomics analysis. Okay, you have understanding about genomics analysis, so that you have understanding about the size of the PCR, and then using target sequencing for final research project. Okay, if you're targeting a particular sequencing for final research project, okay, I think it's a great idea. All right, so okay, I think that's all. Ngazibi, are you still there? Did I answer your question?
Yes, sir. Thank you.
Okay, so I'll probably suggest maybe you can reach out to me, okay, so that we can talk about it. Is that clear?
Okay. Yes, sir. Thank you.
Yeah, you can just chat. Let me open in case if anyone is interested. Let me try to open the group. Are you on the group? Are you on the WhatsApp group?
No,
Ngazibi?
I'm not in the group, but I would like
Okay.
to join.
Okay, just reach out. Just reach out. Just let me send a WhatsApp number. Okay, just reach out to me. This is my WhatsApp number, 771911794. So just chat me up, okay? So somebody said, fantastic opportunity you are providing here, sir. I'm wondering what you take on is using long read sequencing rather than short read for metagenomics. I'm wondering what your take, okay, what is your take, what's my take on using long read sequencing rather than short read for metagenomics? Okay, see, when you use long read, okay, sequencing, most times long read sequencing is focused on an individual, okay, or an individual organisms, okay? Well, there's a technology that goes on working with long read. Just that, you see, the difference is that the long read will be able to enable you to cover more range, okay? Such that in long read, you see, you may have more genes than the short read. I don't know if you understand what I'm trying to say, because these are the advantage actually, the covering area. Because the more a fragmented sequence is longer, the more genes you discover, the more proteins you discover. I hope that is clear, Emeka. Patrick, are you here, Emeka? Hello, Emeka. Are you here? Okay, so if you are, okay, amazing. So that is it, okay? That is it. So, Adelusi, Dr. Adelusi, are you here?
I'm with you, sir.
Yes. Yes.
But I'm not yet a doctor. I'm still a PhD student anyway.
Is that right? I think you dropped something the other time on the chat room.
Come again?
I think you dropped something, maybe like a question or something on the chat room, right? The other time.
Not really. The only question I dropped was regarding our assessment to what's it called, to the video.
Okay. Oh, all right.
Yeah.
No problem. All right. Thank you. So that's it. That's it, everybody. Thank you so much. And I would like to announce to everybody that from next year, probably from January, we'll be having a webinar series. Okay, we'll be having a webinar series that will last for 4 months, and it's free for everybody. It's a free international webinar series we'll be having. We'll be onboarding speakers, keynote speakers from different countries. Okay, those who are professors, who are doctors, who are lecturers, who are experts in genomics field, and we'll be also talking about the career opportunities in genomics, stuff like that. So it's going to be starting hopefully by January, but we'll let everybody know. So if you're on the group, stay on the group. Okay, if you're on the group, stay on the group, because we want to really make impact, and that is providing opportunities to as many people as possible. Okay, so thank you so much. That is the announcement. Thank you very much.
Again, bro.
Great having
Hello,
you guys.
sir.
Yeah. Hello.
Yeah.
Hello. Yeah.
Then, do you guys also try to collaborate maybe with one or two projects regarding the publication aspect of it? Maybe you want to contribute your part, then also towards the publication aspect. Do you guys also do that?
Yes. So you can reach out to us. We'll discuss.
Okay, yeah, I will reach out to you.
Yeah. So that's this. My WhatsApp number is still on the chat room.
Yeah, I think
So
I have your number. I've been following you all this while.
Oh, really? Amazing.
Yeah.
Yeah. No problem. Just reach out. We'll discuss.
All right, I will. Thanks.
Okay. Thank you so much. All right. So thank you so much, everybody. Really appreciate you coming around. See you next time. Have a nice day. Nice time. Thank you.